I'll be going to deal about the monoclonal antibodies, how they have really have the precision therapies in the cancers. The mainly I'll be focusing on the like women cancers. At the same time, I'll be seeing the various other aspects also. So this is my the outline. So some historical foundations and how the production evolution has happened over since last few decades, and what are the clinical applications in the various across the medical conditions, and what are the challenges and solutions, and of course, finally, I will move to the future directions. Monoclonal antibodies have very important like indispensable tools in research, therapy, as well as diagnostics. They are evolved as a magic bullet to the precision therapy, and from this precision therapy, they have been taken part very important role in the broad spectrum of application in the prevention of the various medical disorders, detection and treatment. And if you see the market growth rate, it has been a tremendous, tremendous, and it has been said that from 2024 to 2033, it has been expected to have the compound annual growth rate of around 11.07%. And they have been used for various, not only the non-communicable diseases, including the various communicable disorders also. And till now, there are 145 monoclonal antibodies have been approved from since 1986, from its inception. And in the year alone, 2023, almost of the total new molecules which have been approved, 31 percentage of the total drugs which have been approved in that years, the 31% were the constituted by the monoclonal antibodies alone. So you can see that almost it constitutes one third of all the molecules which have been approved in that year. And till now, there are 91 cancer monoclonal antibodies are currently used in clinics, and there are many in the pipelines, and this is the how it has gone to it. And monoclonal antibodies serves as one of the precision therapy from the magic bullet, revolutionize the treatment landscapes in the cancers, not only the cancer, various other medical conditions where it become the one of the precise therapy, and it has met the unmet needs in various conditions where that condition was not treated with other like other conventional therapies. And the future, of course, it is one of the most promising in the era in the various medical conditions. So coming to what are these monoclonal antibodies? There are specific immunoglobulins, the single clone of origin, and they are very, very, very specific. They are monospecific and homogeneous in their identity, and most frequently used class of monoclonal antibodies belongs to the IgG class, and again in that one IgG 1, 2, 3, 4 are applied with the unique properties. And coming to its composition, it is a large heterodimer proteins with two identical light chain and heavy chains. So I will not be going in that detail. And it has got various regions, and these regions determine the various specificity and its like efficacy, and including the various other aspects to it, therapeutics. And they are highly precise, as already mentioned. They modulate the immune mechanism, long-lasting effects, and have got the many purposes, including diagnostic and the treatment. And this have got various benefit like from the various like use in various clinical condition, including the rare diseases, and have got a rapid effect and has improved the tremendous improved the quality of life, and has been associated with very fewer side effects because of their precise in nature, and they have personalized the approach to the treatment of various communicable and non-communicable disorders, and with minimal drug interaction, with higher specificity and the enhanced efficacy. And this is how the nomenclature says about the it has based on the source, whether it is murine or chimeric or humanized or monoclonal or the human monoclonal antibodies. It could be like OMAB, CMAB, ZUMAB, or UMAB. Antigen selection is one of the critical step in the development of monoclonal antibody. The production is a very complex process, starts with the cell line development, cell culture techniques, and finally purification. It's a big process, involves the various technology. I'll be talking about the other these things. And of course, it has to follow as per the regulatory compliance with a good manufacturing practice. That is the most important thing. That is it should be very pure in nature, and has to be in quality control, and those things has to be followed. It has started from the around 1970s by the Milston and Kohler, who received the Nobel Prize in the physiology and medicine for hybridoma technology, and they developed the hybridoma technology that has led to the complete revolutionize this monoclonal antibody production, and from that onwards, it has been it is tremendously, it is growing the field, this field. And this technology involves the B cell isolation and fusion with the myeloma cells, and cells are produced of desired antibodies which are isolated, and finally it can be produced in the mass production. And as I already mentioned, murine, these are 100% mouse proteins, and chimeric, which has got little part of the mouse, and humanized are mostly human with little bit binding regions from the mouse, and fully human are the 100% human proteins. The first monoclonal antibody which has been approved was in 1986. That was Muromonab, that targeted a CD3 cell and a T receptor blocking, and it was first time it was successfully used in acute transplant rejection. This was the how the story started. But however, the problem with the uh, these murine antibodies are the HAMA reaction, human anti-mouse antibodies reaction, that has led to the various hypersensitivity reaction, and from that one again it has evolved to the from murine to the completely human monoclonal antibodies. And there are various techniques that are for the purifying characterization. I am not going in that detail, and ELISA techniques and other techniques also used. Coming to the mechanism of action, there are various mechanisms have been understood how they will go into. One is the neutralization reaction, binding to the specific target, that pathogens or the toxins it will neutralize. Other second thing is the antibody dependent cellular cytotoxicity, ADCC. This is one very important mechanism, and also the complement dependent cytotoxicity mechanism, and or it could be a receptor blocking. Neutralizing mechanism is simply as it binds antibody binds to the toxins, and it will blocks the, and finally to clear the whatever the pathogens from the body. Antibody dependent cellular toxicity is antibody binds to the specific target cells, FC region, that is constant fragment region, that activate the immune cells like NK cells, macrophages, neutrophils, and finally these immune mechanism, these cells will destroy the cytotoxic substance release they will release a cytotoxic substance which can destroy the target cell. Complement dependent cytotoxicity is again this activated the complement system, and this complement system form the membrane attack complex, which in turn activate the complete the set of complementary mechanisms, and finally which can lyse the target cell. And in between there are various like checkpoint inhibitors have been come across as per the pathogenesis concerned with the cancer. There are various signaling molecules which are involved in the uh, in the cancer cell production, or it could be for the survival cancer cell like CTLA. It could be a program death one molecule is all cytokines. So based on that, specific targets have been identified. These are some examples. I'm just quoting it. Another class set of monoclonal antibodies are the antibody-based agents. Could be there also could be ADCs. That means antibody drug conjugates. They deliver the cytotoxic drugs to the specific targets. Or it could be radio-labeled monoclonal antibodies, which can deliver the radiation directly to the cells, which are the target. And there are also have got something like BiTEs. They are called as bispecific T cell engagers. Coming to this antibody drug conjugate, it has got three important components. One is antibody linker and cytotoxic drugs. So it will exactly target the specific cell and delivers its cytotoxin there. So these are the how it will like it is benefit is reduce the damage to the healthy tissue. So it is very precise. Example like brentuximab, and there are some certain this how it will exactly happen. It targets internalizes the antibody drug conjugate enter in the cancer cell, and it will release the cytotoxic substance to that particular cell and finally causes lysis. And there are with the various approval, these are some examples. And bispecific bites stands for they have got the dual target. It means it bind to two specific antigens which are expressed on the target cell, and finally it will recruit the T cells, which in turn activate the immune mechanism, and finally it will produces its efficacy. And these are the recent like bites approvals in the various cancers, malignant to lymphatic malignant, and other things. Coming to the what was the reactions associated with this monoclonal antibody. The most common adverse drug reaction which has been noted with this monoclonal antibody, the hypersensitive reactions. However, this reaction has been come down because of the use of nowadays more of the humanized and human monoclonal antibodies, and at the same time they are also associated with the myelosuppression because of the blood cell production is reduced, and of course they have the immunosuppression mechanisms and cytokine release syndrome. That is one another serious adverse drug reaction may be associated with this one. And other thing like it could be mild like rashes it's from fever, chills, rashes, some respiratory insufficiency, immune related mechanism. It could be related to any organ specific or like non-specific like thyroid, like colitis, pneumonitis, and other general things. And this is other like adverse drug reactions. Coming to the where are they have been applicable in various cancers, including the various malignancies. You name it, there are monoclonal antibodies, of course, for that particular cancer. It could be any, could be breast cancer, colorectal cancer, esophagus cancer, head and neck, and so many other hematological malignancies and many other solid tumors. And these are some of the examples of hematological malignancies for targeting the various cytokines, and these are the examples for the various monoclonal antibodies. And monoclonal, not only the cancer, they have got various application in the microbiological against various micro viral infection, bacterial and parasitic infection. And not only that one, they are also have got important role took into the part of like various other like cardiovascular like PCSK9 inhibitors like evolocumab, alirocumab, and in the neurology, Alzheimer's disease and migraine, hematology, of course, and various infectious disorders, including the viral like palivizumab and ebalizumab HIV infections, and including, of course, parasitic infection. And there are various diagnostic applications also. They have been have taken important clinical aspect. Coming to the now, I will focus from this onwards. What are the different monoclonal antibodies which have been in the clinical practice, especially against the woman cancer? The coming to the first monoclonal antibody, that is the atezolizumab. It is primarily indicated and approved for the treatment of triple negative breast cancer, and also it is also been approved to treat the cervical cancer. The main mechanism is one of the signaling molecule, that is PDL1. So that is its main target. It inhibits and that is responsible. And it is the patient selection is who are all expressed with the PDL1 positive, and who are all the triple negative breast cancer or the cervical cancer patient. And usually it is administered intravenously, and regular assessment need to be monitored to monitor the adverse drug reaction. And other monoclonal antibody which has been approved for the treatment of cervical cancer is avelumab. Again, it is the target is PDL1 and is again administered bi-weekly. And other monoclonal antibodies includes like bevacizumab. It has been approved for the treatment of various malignancies related to women like ovarian cancer in the advanced stages, cervical cancer, along with the chemotherapy, other chemotherapy drugs, and breast cancer, especially in the metastatic disorders. How this bevacizumab work? The main target of bevacizumab is vascular endothelial growth factor. This is another important cytokine. It binds to that cytokine and prevent the angiogenesis. And as we know, angiogenesis is one of the important event for the cancer cell survival, and that's how it will starve the blood supply to the cancer cell, and finally the cancer cells will go for the lysis. It has been established from the various clinical trials that bevacizumab in ovarian cancer, especially response rate is very high, especially in the platinum resistant cases, almost around 40%, and the median progression free survival is 6.7 months, and overall survival benefit is 16.6 months. And for cervical cancer, it is used in a combination therapy along with other standard chemotherapy drugs, and survival benefits has been well established, and it has been associated with the good therapeutic effect. Coming to the next monoclonal antibody, that is the cadonilimab. It is a dual target antibody. It is a bispecific monoclonal antibody. It target the two important cytokine, that is PD1 and CTLA4 pathways. Both are involved in the pathogenesis of pathogenesis of cancer, and primarily indicated in advanced and recurrent cervical cancers. And it is in the developmental stage, and new antibody is a promising result. And the other monoclonal antibody include plimab. Its target is again PD1 receptor. Has been approved for the treatment of recurrent and metastatic diseases. It is given into the intravenous route. And other monoclonal antibodies which has been there for the treatment of endometrial cancer, that include the dostarlimab. It is approved for the treatment of recurrent and advanced endometrial carcinoma. The mechanism includes PD1 receptor antagonist, and it's the mechanism has been associated with the something called as deficiency in DNA mismatch repair system. That is one of the that can lead to the creates high mutation burden and more immunogenic and more response to the and the cancers which are like having the deficiency in DNA mismatch repair, and they are more respond now got the good response to the drugs like dostarlimab. And coming to the dostarlimab mechanism, it is a PD-1 receptor blockade, prevent the cancer cells from evading immune detection, and finally enhance the T cell attack against this DMMR tumors. And survival rate, overall response rate is around 42%, and disease, it has been said that the disease control is around almost more than 90%, and it has been approved by the FDA in 2021. And coming to the next monoclonal antibody, that is the durvalumab. It is approved in 2017 for the treatment of the bladder cancer and as well as the other malignancies. And the target is PDL1 receptor. This is also has been approved for the treatment of cervical cancers, and in some countries it has been approved, and the still trials are going on, and has shown a very promising results with a emerging efficacy data. Other monoclonal antibody include irinotecan. It is a trifunctional monoclonal antibody targeted against breast cancer cells, and this binds. This main mechanism is it binds to the HER2 receptors, which attaches to the cancer cell surface, recruit the T cells, engaging the CD3 on T cells, and activate the immune mechanism, and finally destroy the cancer cell. That's how it will work. And again, it has been said that. And the next other drug is like margetuximab. It is also another drug which will works on the HER2 receptor, and it has been approved in 2020 for the breast cancer. Another drug which is acts on this HER2 receptor includes margetuximab. It has got an enhanced immune response and primarily indicated for the treatment of metastatic HER2 positive breast cancer cells. Other monoclonal antibodies include mirvetuximab. Mainly, it is uh, has been approved for the treatment of platinum-resistant ovarian cancers, and it delivers cytotoxic payload directly to the cancer cells. And this platinum-resistant ovarian cancer cells, by definition, it is a recurrence of the malignant cells within 6 months after platinum therapy, and typically it has got poor prognosis and and limited effective options. However, this drugs like mirvetuximab has been found to be have got the efficacy in these kinds of settings. How mirvetuximab works? Again, it targets this receptor called folate receptor alpha on cancer cells, and cause internalization of the cancer cell with the antibody drug complex enters the cancer cell, releases the cytotoxic polyload, and finally disrupt the cell division and causing the apoptosis. Other drugs include nivolumab. Again, it is a monoclonal antibody approved for the treatment of cervical cancer, targeting PD-1 receptor, and also it has been used for the treatment of recurrent and metastatic disorders. Other include like pembrolizumab. It has been mainly approved for the treatment of cervical cancer, endometrial cancer, and the breast cancer, especially the triple negative breast cancer. Its target includes PD-1 receptor, prevent the deactivation of the cancer immune suppressing signals, and activate the T cells, and finally uh, destroy the cancer cells. Pembrolizumab has also been approved for the endometrial cancer, especially for the MSI or the DMR, as I already mentioned, with the which have got the DNA mismatch repair, and response rate is around 50%, and treatment duration has to be continued for 2 years. Pembrolizumab in breast cancer, it is approved for the triple negative breast cancer, PD-1 receptor antagonist, and is also been used along with the conventional chemotherapeutic drugs. Other drug includes pertuzumab, primary indicated in the breast cancer cells, specific type. Its target is HER2 positive breast cancer cells, often combined with the trastuzumab. And this by targeting HER2 receptor bind to specific its domain, and blocks this pairing of HER2 and HER3, and finally that carries the signals, and that stop the growth signals, and leading to the uh, lysis of the cells, cancer cells. This is a and the almost median survival rate is around 56.5 months, and free survival benefits are almost 18.7 months, and response rate is around 80%. The next monoclonal antibody for the against the breast cancer cells include sacituzumab. It is approved for the treatment of metastatic triple negative breast cancer cells and the antibody drug conjugate, and it targets the prop2 antigen of the cancer cells. And by definition, triple negative breast cancer cells are which lack the estrogen, progesterone receptors, and HER2 protein, and they are more aggressive with fewer treatment options. And the drugs like monoclonal antibodies have revolutionized the treatment of these triple negative breast cancers, like atezolizumab, sacituzumab, and pembrolizumab. Coming to the other, the mechanisms with this sacituzumab includes targeting to the on cancer cells. This complex enter the cancer cells, and finally it will deliver the cytotoxic load inside the cell, and finally the cancer cell is destroyed. Other monoclonal antibody include socazolimab, which is target the cervical cancer through the anti-PD PD-L1 receptor, and it is in the clinical trials. Tisotumab, it is primarily indicated in cervical cancer and is the antibody drug conjugate which delivers the cytotoxic load to the specific cancer cells and target the tissue factors. And it has been the free survival period has been found around 4.2 months. The other monoclonal antibody, trastuzumab, has been targeting the HER2 positive receptors, and they have been expressed in the breast cancer cells, including the endometrial cancer as well as the ovarian cancers. HER2 positive breast cancer. Uh, um, these are the breast cancer cells which have got the overexpression of HER2 receptor proteins are around the cancer cells, and it has been said that approximately 20% of breast cancer are having this HER2 receptor positivity, and the various monoclonal antibody which have been found to have their therapeutic efficacy against this HER2 receptor positive in breast cancer include trastuzumab, pertuzumab, margetuximab, and ertuximab. Trastuzumab, as already mentioned, it is a HER2 receptor against this cytokine, and it stops the growth signals and activate the immune system, triggering the antibody-dependent cellular cytotoxicity, resulting in the cell lysis. For breast cancer, it was the first approved pioneering HER2 targeted therapy, and it requires the monitoring with the cardiac cell function. Uh, and trastuzumab is the endometrial cancer is indicated for HER2 positive endometrial cancer, less common but effective in selected cases. It is also then coming to the various monoclonal antibodies which have been approved for the treatment of endometrium include dostarlimab, pembrolizumab, and trastuzumab. Coming to the cervical cancer treatment, the monoclonal antibodies include bevacizumab, pembrolizumab, isotumab, and semiplimab, and targeting the various cytokines like VEGF, PD-1, tissue factor, and PD-1. And coming to the monoclonal antibodies which have been approved for the treatment of ovarian cancer include bevacizumab, mirvetuximab, and trastuzumab, and targeting similar like VEGF and the HER2 receptors. This is a table showing the how the monoclonal antibody that means the various cancers cells targeting the various receptors or cytokines and their uh, uh, applicability in the clinical practice, especially in the malignancies related to the humans. Coming to the what are the problems associated with this monoclonal antibody? Similarly, like other conventional chemotherapy, this monoclonal antibody also have to go for the resistance mechanisms, and this is because of various reasons. Number one is the antigen loss, down regulation, or the mutation of the target could be the one reason, and another phenomena called trogocytosis. It is nothing but transfer of membrane fragments between the cells, or the complement exhaustion is another reason for the resistance, or it could be immunosuppression. To overcome this resistance, there have been the various strategies um have been followed. One is need to understand the underlying mechanism, or the how why the resistance is happening. Second thing is we need to have the multiple combination approach, similar like other like against the microbial infections. In the similar way, the multiple antibodies increase the effectiveness. That is the one way, and the multi-antigen targeting, like bispecific monoclonal antibodies. Nowadays, trispecific monoclonal antibodies have also been have been in the pipeline. They will target the different antigen epitopes present on the cells, and by that acting by through the multiple mechanism, and this is another way of like escaping the resistance. And adaptive therapy is responding changing to the as per the tumor biology. And these challenges and other limitation includes immunogenicity. As already mentioned, it was very high with the murine type of monoclonal antibodies. And of course, definitely cost and accessibility is one very important factor that always come when you come to the monoclonal antibodies. And resistance, as already mentioned, and tumor penetration, some especially the solid tumors. And however, there are a lot of research, and there are various monoclonal have been come across that have been improved with their stability, including their permeability through the various solid tumors. And these challenge also include like the complex pharmacology requires further understanding the not only the pharmacology, physiology, and pathology, how exactly this the cancer cells are like various signaling molecules involved in its pathogenesis. And of course, there are definitely regulatory hurdles, especially for the global accessibility and logistic consideration need to be taken care of. To address the challenges, there are one this thing is human monoclonal antibodies, of course, reducing the immunogenicity through the fully human monoclonal antibodies. That is one thing. It has already there are various fully human monoclonal antibodies are in the clinical practice that have reduce the hypersensitive reactions. And second thing is coming to the cost. The cost and accessibility has been taken care by the biosimilars. And the first biosimilar which was approved was the rituximab in 2018, and with a very lower cost when compared to it's like the reference compound or the novel originator, and it has increased the accessibility across the globe. And another thing is something called as biobetter. These are nothing but engineered improvements over the original antibodies, enhance the efficacy and reduce the adverse drug reaction. This is also one thing that I'll be talking about a little bit something about the biobit in the next slide. And of course, the combination therapy is the one strategy to overcome the resistance through the multiple mechanism. Biosimilars are the highly similar versions when compared to the approved monoclonal antibodies are the originator and have reduced the cost, and they are affordable and at the same time accessible around the globe, and increase accessibility and similar almost have the similar efficacy when compared to the originator molecule. Biobetter are the engineered improvements over original monoclonal antibodies, enhance the efficacy by improving the binding or the functional properties, and have been reduce the various adverse drug reactions related to monoclonal antibodies, especially these hypersensitive reactions and the infusion related reactions, and have got improved delivery to the specific targets. And coming to the, I'll be talking a little bit about what are the future, the research direction, especially in the monoclonal antibodies era, and the production innovations are it is going in a very fast, rampant way, and there are various novel engineering approaches have already been in place, expanded application, including the various uh, like multidisciplinary team and the common strategy has been in place, and there are various novel formats have been designed by the bioengineers with the help of the various chemists, including the clinicians, and AI integration is already in place for the computational design and optimization. And for the sustainable production, there are plant-based and alternate systems also have been in the process of exploration, and global accessibility by affording the manufacturing solution. The next generation platform, of course, definitely includes cell-free expression. It is eliminating the cellular limitations. Continuous manufacturing can be done with this through these things. And novel antibody formats includes not alternate to the antibody class, not only the IgG, the most commonly the immunoglobulin used in the monoclonal antibody production IgG. Now they are going for the other alternate other than the IgG like IgA, Ig, and IgM. And single chain antibodies also in the pipeline simplified, they are having simplified structure with a maintained binding capability. And catalytic antibodies are the one monoclonal antibody with enzymatic properties, and transgenic mice platforms are the one which are reliable human antibody development without humanization. That is also is another novel antibody format. And future is of course definitely beyond the IgG molecules with the single chain antibodies, catalytic antibodies, and the novel architectures. And not only that one, there are various novel new antibody drug conjugates also in place, like nanoparticle conjugate, catalytic toxin conjugate, and novel payload conjugates. And other like I have already mentioned the bispecific monoclonal antibodies. Now there are also something called as tri-specific monoclonal antibodies, which engage the three targets for even more complex interaction. And nanobodies are the smaller antibodies format with unique penetration ability, especially the solid tumors. And already mentioned tri-specific antibodies, they target three important regions on the cells of its target, like enhance the immune engagement, especially CD3, CD28, and T cell activation. It is one of the emerging technology moving from lab to the clinical application and have got many potential advantage in the form of greater specificity and efficacy possibility. Nanobodies are the one other. They have the single domain monoclonal antibodies smaller than the conventional one, derived from heavy chain only, and they have got advanced with the better tissue penetration, stability, and the production efficiency. And they are have been applied and explored in the diagnosis at the same time the treatment, including the African trypanosomes. And neoantigens are the one other that is another concept is coming. This is based on the target specific to the cancer cells. As the cancer evolves and as the targets goes for the mutation, and based on that one, these antigens, the most common one are like BRAF, RAS, and TP53 have been identified, which probably most probably they will go for the mutation and leading to the failure of or the resistance of these monoclonal antibodies. And the new antigens are the one which will target specific to this cytokines, and that's how it will bring down the enhance the efficacy, and at the same time it comes down the resistance pattern. And there's something like glycoengineering, the one thing where the constant fragment of the monoclonal antibody is modified with alteration in the glycolization process that has enhanced the affinity toward the various immune cell mechanisms, and finally which will enhance the ADCs. That is the one other concept is already it is there, and no other conjugate I already mentioned. There are various. It could be a radio labeled, or it could be a nanoparticle, and many others. There are various examples I can say. Ibritumab, that is like a radioisotope conjugates, have been applied in the various malignancies, including mainly the lymphomas. And artificial intelligence already it is in place, which has taken its important role in in the discovery and in the making the process faster and to understand in a better way. Target identification with algorithms predict the various optimal targets, and machine learning can improve the antibody designing, and of course, and to also predict the various analytics to enhance the testing, the clinical trials, and of course in the target selection, the treatment, the clinical practice. And there are FC engineering, protein engineering, electrophoresis. There are various advanced production techniques also in the pipeline, and there are various other like advanced development include B cell isolation, plant-derived antibodies, and artificial intelligence. And transgenic mice is the one thing which express the human IgG isotypes similar to human thing, and that is one place. And plant-derived monoclonal antibodies it is explored as the alternative model for the low-cost oral delivery potential, and the next generation structures include like the that is a small chain like FE and VHS based single variable domains, and transgenic mice platforms another one where they have been like trying to have the same sim like monoclonal and single B cell isolation is another technique there. It is already there for the production of the various monoclonal antibodies, and various other advanced engineering techniques already has been explained this one. And gene edited antibodies are the ones, CRISPR technology, optimized binding, and other things already is in the place to personalize therapy still more in a better way. mRNA therapeutics, nucleic acid encoded, and the gene transfer technology already in the place to target this as per the more in a precise manner to target the specific uh, specific cytokine for the pathogen. And antibody plus that already there is another one thing that already we know the CAR T cell therapy, that is chimeric antigen receptor T cell, there are living drugs, and this TCR like antibodies are the one generate the CAR T cells using variable regions, and integrating these antibody components with other modalities has been treatment for various like especially hematological malignancy, and this is one of the major breakthrough in the advancement of the cancer immunotherapy, and it is already the monoclonal antibodies are like combining, they are combined with the various like CAR-T cells to enhance further efficacy against the various malignancies. And plant-derived antibodies, I already mentioned, these are expressed with the plant systems and prevent the treatment of various infectious disease, and they have what said to have the low cost production when compared to, as that's the one important uh, uh, aspect related to the current situation, because they are very costly, and that is the one reason they are trying to explore in the different system. And there are these are the various fragment based approaches like fab fragments, SCFE, nanobodies, diabodies, based on they have got various advantages in the tissue penetrability, and they're specific to the particular cells. And of course this already has been mentioned. And various like the era is the combination with the various not only the monoclonal antibodies, with the chemotherapy drugs, various checkpoint inhibitors, small molecule like monoclonal antibodies, finally to attack in through in a holistic manner to the cancer cells, and finally making the better efficacy against the cancers. I mean, this is how the clinical pharmacy have played very plays very important role in like optimizing the dosing PKPD principles, including its predictive efficacy, the multidisciplinary team, and next generation monoclonal. Of course, definitely they will enhance the precision, multi-targeting, simplified manufacture, and extend the half-life. To conclude, they are the precise weapons as a magic bullet, specific to that to a particular target, have got wide applicability in the various, not only the cancers, across various medical conditions, including communicable and non-communicable disorders. And continuous innovation is it is in the can address the various challenges which we have fit fast till now, and definitely multidisciplinary approach will can definitely give the lot of solution. And these are the various like key biomarkers, especially when it comes to the women cancer, like HER2 receptor positivity, PDL1 status, and the DNA mismatch repair, and folate receptors. And this is a summary of various monoclonal antibodies already in the clinical practice to treat various cancers, including breast, ovarian, and cervical cancer. And and key takeaways is monoclonal antibodies have got a significant evolution, advanced the completely. They have made the very important role in taking care of the treatment of the cancer, especially which are not treatable with the conventional chemotherapies, and have got many clinical benefits because of their highly specific in nature, with a very minimal drug interaction and the minimal adverse drug reaction. And there are various formats are already in the place, like bispecific antibody drug conjugates and various glycoengineering, and many technology based designing is already in the pipeline to further enhance their efficacy, and continued advancement is definitely with the next future. With this one, I will stop my uh, speech, and thank you very much.